Welcome everyone to BPL Nepal. Today we will going on the MCQ related theory of Plasmodium Vivax. I think it will become of one or two questions or maybe four questions. Okay, let's start. Firstly, we will detailedly study about Plasmodium Vivax. I think it will take 15 to 20 minutes, and I have included today. I am not able to present digitally as well as in board, so I am sharing my notes to you. I hope you like it, and I have summarized the points in very simple manner. Let's start. Okay, let's see. Plasmodium vivax. Plasmodium vivax name was given by Grassi and Filati scientists were Grassi and Filati who gave the term Plasmodium vivax. It was first discovered by Charles Lavrin. As well as the discover of female Anopheles mosquito is agent by Sir Ronald Ross, who was a military doctor. Sir Ronald Ross. There are three scientists that you should mainly remember. Firstly, this is very important. Yes or not? Causative agent, which is the causative agent, who discovered the causative agent that female Anopheles mosquito is the causative agent for the Plasmodium vivax or transferring material, means Sir Ronald Ross. And who named Plasmodium vivax? Grassi and Filati. And who discovered Plasmodium vivax? Charles Lavrin. Okay, this is also main important. Let's see classification. Kingdom Protista, phylum Protozoa, class Sporozoa means locomotory, there are no locomotory organs. Order Hemisporidia. Hemi means blood sucking. Hemisporidia means blood sucking. Family Plasmodium. Genus Vivax. Plasmodium is best known as intracellular, means digestion is inside the body. Digenetic means two host. Protozoans they are. Endoparasite means inside the host animal. Endoparasite of man, which causes malaria. They are the causative agent for malaria. There are three types of Plasmodium. Mainly three types there are. One is Plasmodium vivax, Plasmodium ovale, Plasmodium malari, and Plasmodium falciparum. Plasmodium vivax, Plasmodium ovale, Plasmodium malari, Plasmodium falciparum. Let's see. Habitat. It is a common endoparasite of man, which is usually found in liver and blood of man and salivary gland and stomach of female Anopheles mosquito. There are two stages, trophozoite and sporozoite, feeding stage and infective stage. They are found in gut of stomach, salivary gland of stomach, as well as RBC of man and a liver of man. There are different stages. We will detailedly study later on. Likewise, let's see structure. They are dimorphic. They are not polymorphic. They are dimorphic. They have two different stages, so they are dimorphic. And the most important two structures are trophozoite and sporozoites. I have mentioned. You can see the examples or figure. Let's see. First figure is of, this is trophozoite. What this is? This is trophozoite, means which is found in RBC of human, RBC of blood. Let's see. We have to detailedly study about this is the feeding stage. Okay, this is trophozoite. Let's see. Outer layer is covered by double layered plasma lemma. Let's compare. In comparison we will look. Okay, this is sporozoite. Sorry, this is trophozoite and this is sporozoite. This is feeding stage and this is, one is feeding stage, another is infective stage. Let's see. Body is covered with plasma lemma in trophozoite. In sporozoite, body is covered with single layered pellicle. They contain nucleus. They also contain nucleus. They contain concentric ring. They does not contain concentric ring. They contain food vacuole. But it is not mentioned. They have cytoplasm, mitochondria. They have hemogen. In food vacuole. This is food vacuoles. These are food vacuoles, but they does not contain hemogen. Okay, they does not contain, because they digest hemoglobin and relate the hemogen, but they does not digest. They have microtubules. Which is used for penetration of liver cell. For penetration they use microtubules. Mainly no, not microtubules. There is here is the pure organs that release proteolytic enzyme which dissolve the protein layer of liver cell. They release proteolytic enzyme which dissolve the first layer of liver. Okay, they contain Pladase granules is protein synthesis that does not contains Pladase granules. Okay, you can take a screenshot of these two pictures and you can read. Mainly you have to understand that they contain plasmalemma, and you have to remember this hemogen and in Pladase granules and concentric ring. Likewise in here you have to remember microtube. Body is covered with, this is not this much, only half part is not covered, all the whole part of the body is covered, okay, by microtubules. They have apical cap, and main important is pure organelles that release proteolytic enzyme. You could be asked what type of enzyme is released by the pure organs to penetrate the liver cell, then means proteolytic enzyme to dissolve the layer of liver cell. Okay, let's move on. I have mentioned the difference between, it will be easier for you as well. Let's see, trophozoite and sporozoite. Trophozoite, this figure, sporozoite, this figure. Let's see, it is feeding stage. Trophozoite is feeding stage and sporozoite is infective stage. It is, if it is feeding stage, then it must be found in RBC of man, where is hemoglobin. Likewise in sporozoite, it is salivary gland. It is found in salivary gland because from female Anopheles mosquito it is it fluctuates to the liver cell. Okay, it inoculates to the liver cell. In trophozoite, let's see, trophozoite is spherical in shape, and sporozoite it is elongated nail-like in shape. Likewise, in trophozoite, body is covered by double-layered plasmalemma. Trophozoite body is covered with double-layered plasmalemma. In sporozoite, body is covered by single-layered pellicle. Here plasmalemma, here pellicle, and large number of food vacuoles with hemogen pigment. Absence of food vacuole. Both SER and RER are present, okay, in trophozoite, but in sporozoite only endoplasmic reticulum is present. Their types is not present. And in trophozoite, let's see, presence of concentric body. Here absence of concentric body. Absence of pure glands because they need not to penetrate any liver cell, but here presence of pure glands. Here Pladase granules help in protein synthesis in place of ribosome if ribosome are absent, but here Pladase granules are absent. Main important is wriggling movement. You have seen that there is a long tail-like structure, that is microtubules. Microtubules help in wriggling movement. Wriggling movement means like this movement. Wriggling movement means like this movement. Okay, wriggling movement and microtubules are absent. Whole body is covered by microtubules and have wriggling movement. This much for two stages. Let's study detail on the life cycle. From life cycle you will be asked many more questions. Let's see. First we have understand that these are digenetic parasite. Digenetic endoparasite means digenetic hemo-endoparasite. Okay, then they should have two hosts. One is, if there is asexual reproduction in that organism, means that is secondary host or intermediate host. If there in those hosts in which asexual reproduction is done, then that is called secondary host or intermediate host. And if there is the presence of sexual reproduction, then you have to understand that that is primary host or definite host. Let's see. First, asexual life cycle of plasmodium. Life cycle of plasmodium in men or secondary host or intermediate host. First you have to understand the term inoculation. What do you mean by inoculation? The process of releasing large number of sporozoite Plasmodium vivax in the human body by female Anopheles mosquito called inoculation. Means release of large number of sporozoite into the body of into the body of human by female Anopheles mosquito is simply called inoculation. Likewise, let's see, this sporozoite which is released into the body circulate about half an hour, then only enter to the liver. Okay, they circulate in a blood for half an hour, they circulate in a blood for half an hour, then only they enter into the liver cell, starting liver cytogony. Now you have to understand that what do you mean by liver cytogony. Cytogony is simply, let's see, it is a type of division in which nuclear division completed first. Then cytoplasmic, it is a division in which nuclear division completed first. First nuclear division completes, then cytoplasmic division occurs. If takes place in liver is known as liver cytogony, or you cannot write this liver, then it is cytogony only. Cytogony means it is a division in which a nuclear division takes first, then the cytoplasmic division occurs, then it is called liver, then it is called cytogony. Let's see more. After about half an hour of circulation, sporozoites enter into liver and reproduce by cytogony. During this period, sporozoid disappears in blood. So it is difficult if there is a cytogony, liver cytogony, then for 8 days you will not found any Plasmodium vivax in blood because they disappear in blood. Okay, we cannot detect and don't appear before 8 days. It includes the following stage: pre-erythrocytic, post-erythrocytic, then exoerythrocytic. Okay, let's see. First is erythrocytic phase. It takes about 8 to 10 days and release about 10 to 12,000 of cryptozoites. This is one liver cell. This is liver cell. Okay, sporozoid is entering into this. Then next is cytogen stage. After this, they start to collect some cytoplasmic strand and make a cytogen structure. Then they divide meiotically and make many nuclei. Then merozoites are found by attaching some amount of cytoplasmic materials around them, and cryptozoites are found. Let's see, in 8 to 10 days to produce 10 to 12,000 of Plasmodium vivax in pre-erythrocytic phase. Let's see, another is exoerythrocytic phase. Again the cryptozoites that are released from the pre-erythrocytic phase attacks to the liver cell, and this is known as exoerythrocytic phase. In exoerythrocytic phase, the Plasmodium vivax called cryptozoites released are again invade the liver cell. It takes for 4 days. Then again cytogen. Then here we have wrote merozoites. Okay, merozoites. Here we will write cryptomerozoites because they are the merozoites of cryptozoites. So cryptomerozoites. Then they are called mega metacryptozoite. Then last stage is known as one is they are simply called metacryptozoite. Here in last stage you will get two varieties of cryptozoites. They are metacryptozoites. One is mega metacryptozoite and micro metacryptozoite. In zoology you have to understand that male are smaller, means micro, male are micro, and female are mega. Okay, female are mega, and if there is any condition of attacking anything, then only male are active. Okay, only male are active. Then what? And in next stage, the male one, micro metacryptozoid, will invade the RBC in erythrocytic cytogony. Okay, in erythrocytic cytogony, micro metacryptozoid will invade. Okay, you have to understand this some related terms, like first we have got cryptozoites. Again it is attached to the liver cell, cryptozoites. Then cytogen stage is same. Then many nuclei is formed. Then here we called it is merozoite, and here we will call it as cryptomerozoite. Then again it is bursting, liver cell is bursting. Then two types of cryptomerozoites are formed. One is mega metacryptozoite and another is micro metacryptozoites. Micro metacryptozoites attack to the goes to the blood stream to attack to the RBC and mega cryptozoites again invade the again invade the liver cell and form the post erythrocytic phase. First we will detailedly study on erythrocytic phase cytogony. I have seen many MCQ related problems. Let's look over erythrocytic cytogony. Okay, let's see. In erythrocytic cytogony, what sort of stages do you find? Let's see. This starts from this micro meta cryptozoites attack to the RBC. You cannot write here like this because in RBC we don't have nucleus. Okay, we don't have nucleus. Let's see. Micro meta cryptozoites first goes to the trophozoite. Trophozoite means feeding stage. Okay, trophozoite means feeding stage. It feeds a lot of hemoglobin and it pulls the nucleus towards the one end. Okay, it pulls the nucleus towards the one end because the food vacuoles are more. It eats the hemoglobin, and this stage is known as signet ring stage because it is ring like stage. Okay, यो के कस्तो स्टेज छ त भन्दाखेरि रिङ लाइक स्टेज. इट इज लाइक रिङ स्टेज. ओके, इट इज लाइक रिङ स्टेज. Likewise moves on. In moving on, now see, there is flagellation like structure which is known as amoeboid stage because the Plasmodium vivax release the enzyme that digests the hemoglobin and size gradually decreases. In digestion process we will see amoeboid stage. Here we can find soft knots dot is also present in amoeboid stage. Okay, soft knots dots means like that dots are present which is also found in amoeboid stage. Then it enters to the schizoid stage. From schizoid stage there is not more nuclei, only 12 to 24 nuclei are found, and let's see, there the hemoglobin is digested into hematin, is digested and differentiated into hematin and hematin and globin. Globin is used as protein and hematin is formed into a toxic substance called hemogen, and main cause that we get malarial fever in 48 hours because for whole this process we need almost 48 hours. Okay, we need almost 48 hours. We need almost 48 hours. So the malarial fever occurs in 48 hours. In other paramecium, other varieties of plasmodium, we get from 72 hours and 24 hours. In Plasmodium falciparum you get 28 hours after 28 hours, and in Plasmodium malaria you get almost 72 hours. Fever repetition is 72 hours fever repetition. Likewise, let's see, in hemoglobin is divided into hematin and globin which is used as protein. Hematin is formed as a toxic substance called hemogen. Okay, hemogen, and you have to remember that only 12 to 24. Okay, not more than this, only 12 to 24. Then merozoid stage is formed. This is called a rosette state because here you can see here are hemogen and they are arranged in rose like structure. Rose petals, if you have seen rose petals, then you will see like this structure they are arranged. Now it burst on, and there are two gametocytic stages. One is mega gametocyte and one is micro gametocyte. I have always said you in this video that micro is male, they are active, and mega is female. Okay, let's see, liver. Now this mega gametocyte goes to liver again to attack, and this both stages are directed or shocked by the female Anopheles mosquito to occur sexual reproduction. Okay, to occur sexual reproduction. This mosquito produce the enzymes which digest all the other stages, but they cannot digest megagametocyte and microgametocyte. Nature is like this, okay? We cannot say for nature is great, so we have to respect nature. It could produce other enzyme that could digest, but if they are digested, then the further process cannot be done. So they release such type of enzyme that cannot digested megagametocyte and microgametocyte. Don't be confused on megagametocyte, microgametocyte, and mega metacryptozoite and micro metacryptozoite. Okay, micro metacryptozoites. This is all thing. Okay, you can take one screenshot of this. Let's move on. Now, sexual reproduction, formation of gametocyte. Let's see. After many generation, in this you could ask one question like, what is the incubation period? Means first appearance of symptoms. In how many days you will get first appearance of symptoms? Means in almost 14 days in paramecium caudatum. In paramecium caudatum, in 14 days you will see after the enter of the body, after the plasmodium enters to your body, you will get 14 days symptoms. 14 days, in 14 days, 12 to 14 days, 12 to 14 days. Because why? You have to remember all the processes. Okay, के भन्दाखेरि, let's see, in pre-erythrocytic phase is completed in 8 to 10 days. Yes or not? Pre-erythrocytic phase is completed in 8 to 10 days. Likewise, exoerythrocytic completed in 4 days, and erythrocytic phase is completed in 48 hours. Means 8, 4, 12, 13, 14 days. Means almost 12 to 14 days you can write. Okay. Likewise, let's see, formation of gametocyte. Formation of gametocyte, we have taken two gametocyte stages. One is microgametocyte and megagametocyte. After many generation, in about 4 to 5 times of erythrocytic zygogony, not one time, in 4 to 5 times of erythrocytic zygogony, you have to understand that this micro metagametocyte only attacks the young RBC. Okay, only attacks the young RBC in Plasmodium caudatum, but in Plasmodium falciparum, that is deadly, that attack the old, injured, all type of RBC. Okay, all type of RBC, so they are deadliest. Formation of gametocyte, after many generation, in about 4 to 5 times of erythrocytic zygogony, large number of RBC are destroyed and causes anemia. In such condition, some merozoites don't take part in erythrocytic cycle and they change into gametocyte. Okay, they change into gametocyte. They are megagametocyte and microgametocyte. Megagametocyte have large cytoplasmic material, large cell size but small nucleus, kidney shape. Okay, kidney shape. And microgametocyte have a small size but large nucleus. Likewise, let's see now on sexual reproduction of Plasmodium. Life cycle of Plasmodium in mosquito, female Anopheles mosquito, primary host and definite host. When a healthy female Anopheles mosquito bites an infected person and sucks blood, then the gametocyte and other stages of Plasmodium are released to stomach of mosquito. Blood and other stages of Plasmodium are digested by digestive enzyme, but gametocytic stage remains unaffected. But gametocytic stages are not digested. Okay, and there is the formation of 3 stages. One is gametogenesis, fertilization, and sporogony. One is gametogenesis, fertilization, and sporogony. This is the whole process. Let's see. This is the whole process. First, we have gametogenesis. Gametogenesis include from this to here. From here to here. This is gametogenesis. Okay, gametogenesis means formation of gametes. Then from here, this is fertilization. fertilization. This stage is fertilization, formation of okinite and oocyst. Okay, then here this stage is sporogony. I have mentioned in very short. Okay, let's see. From erythrocytic zygogony, you have got two types of two types of gametocyte. They are microgametocyte and megagametocyte. Microgametocyte is double layered with large nucleus. Megagametocyte is double layered with single nucleus. Let's see. First see female part, or let's see male part first. Microgametocyte, first it is age same it is, then it shows many nuclei, and many nuclei, then each nuclei is covered with cytoplasmic material to form flagellation like structure. So if you ask in which gametocyte X flagellation occurs, this is X flagellation. What it is called? X flagellation. In which X flagellation? Then microgametocyte means male. In male there is X flagellation. Okay, in male there is X flagellation. Likewise in gametocyte, megagametocyte, you can see it divides first into 2n number of chromosome, then again divides and n number of chromosome, and 3 disappears as a polar body. Here were 3 nucleus, but 3 disappear as a polar body, and it form. This process is known as oogenesis. Fertilization or a formation of development of microgametocyte is known as spermatogenesis, and development of megagametocyst site is known as oogenesis. Likewise, these two fertilized with each other. This is known as reception cone. Yes or not? Reception cone. This is known as reception cone or fertilization cone also you can say. These two fertilized with each other. This is megagametocyte and this is microgametocyte. Megagametocyte and microgametocyte. Okay, this form into an untouched, unmotile stage okinite. Then this is the, let's see, this is the stomach. Okay, this is the stomach, and they invade into the stomach wall. They invade into the stomach wall. Then this is not necessary. Then this is converted into double layered stage called oocyst. Double layered stage called oocyst. Okay, motile stage. Then they divide to many nuclei to sporozoid the burst, and they go into salivary gland. Okay, then you can say where does the sporozoid found? Sporozoid can be found in salivary gland as well as liver cell. Okay, as well as where? Liver cell. Likewise, let's see, in mosquito, whole sexual cycle is completed in 10 to 12 days. Okay, in mosquito, whole sexual cycle is completed in almost 10 to 12 days. Here are some extra points. Incubation period means the interval between incubation of sporozoids and first appearance of symptoms is called incubation period. In Plasmodium caudatum, there is 14 days of incubation period. Likewise, let's see, auto-infection. What do you mean by auto-infection? Merozoids liberated from RBC are destroyed by WBC or phagocytic cell, while other attack RBC and whole erythrocytic cycle is repeated again. It's called auto-infection. You can read this by yourself. Okay, now let's study types of malaria. You could ask which is the deadliest. Okay, which is the deadliest and which is the mild. Deadliest is Plasmodium falciparum. Okay, let's see, and minor is Plasmodium malariae. Let's see, question can be framed in this only. Okay, types of malaria: benzene, torten or vivax malaria. Benzene, torten or vivax malaria is caused by Plasmodium vivax, which is The fever repetition period is 48 hours. In 48 hours fever repeats. Okay, and again is mild totten or oval malaria. Here is totten means problem like, and mild totten means simple problem like. Benign totten or Vivax malaria. Benign totten or Vivax malaria. Benign totten or Vivax malaria. Let's see. Benign totten or Vivax malaria. Benign totten Vivax malaria. Benign totten Vivax malaria caused by Plasmodium vivax, which repeats fever in 48 hours. Likewise, mild totten or oval malaria. Mild totten or oval malaria. Mild totten or oval malaria. Plasmodium ovale for 48 hours. Likewise, malignant malaria. Malignant malaria, deadliest. Malignant means dangerous malaria. Plasmodium falciparum. Plasmodium falciparum, which fever occurs in 28 to 48 hours. Likewise, another is quartan malaria. Plasmodium malaria. Quartan malaria, Plasmodium malaria. You have to understand, one is benzene totten or Vivax malaria, which is caused by Plasmodium vivax for 48 hours. And there is mild totten, which is caused by Plasmodium ovale, which repeats fever in 48 hours. Likewise, in malignant malaria caused by Plasmodium falciparum, in which fever repeats in 28 to 48 hours. And another is quartan malaria, in which fever repeats in 72 hours. Plasmodium malaria. Let's see symptoms. I think you no need of symptoms. Then also see, malaria is characterized by alternate fever, sweating and chilling. Fever repeats after 48 hours. Fever increases up to 104 to 106 degree Fahrenheit. Nausea and vomiting. Tongue may be sticky. Coated anemia may occur due to the destruction of large number of RBC. Likewise, liver and spleen get enlarged. Okay, liver and spleen get enlarged. Generally, patient becomes weak and feeding headache. Treatment is also asked. Some of the question I have found that they were asking on the medicine. Let's see. Medicine are, first is quinine, chloroquine, paludrine, resocin and plasmazine. Quinine is oldest. Quinine is oldest. Quinine is oldest medicine that has been used. And paludrine is superior. It can destroy all stages. Likewise, resocin is most used. Quinine, chloroquine, paludrine, resocin and plasmazine. Okay, this much for today. I think, thank you for looking BPL Nepal. And if you need the personal notes, then you can go on my Telegram group. There you can personally chat with me. Okay, if you like it, then give compliment or what I should improve or furthermore. And if you like it, share. Rather, I don't compel you to share. Okay, thank you.